everyone. I want to thank the organizers of Canola Week for inviting me to give an update on our seed degreening work with canola. I want to thank Rex and Sally for this invitation, and also to Lindsay for setting this up. So today we'll be talking about genetic manipulation of ABI3. Yeah, ABI3 is a transcription factor, ABA-inducible transcription factor, and now it confers frost-tolerant seed degreening in canola. So first off, there's a number of research projects going on in the lab. So we have fundamental work going on, looking at reproductive signaling in plants. So we look at farnesylation and hormonal regulation of farnesylation in using our ABI3 as a model system. We look at hormonal crosstalk between AB and BR and how it can influence drought tolerance. We do custom gene editing using either the CRISPR-Cas9 system or the MAT7 system both for research and also for clients. And we also look at different ways through which we can actually improve shatter tolerance in canola. So for today's talk, we are going to be looking at the canola degreening problem. So what is the canola degreening problem? When canola matures under normal development, when there's no frost, the embryo loses all its chlorophyll and it becomes pale yellow. When you press it, you get high quality oil. But when the same embryo, when it's hit with frost around 22 days after pollination, you get the green color being fixed in these embryos, and they can't decolor anymore. When you press this press the seeds for oil, the chlorophyll leaks into the oil, and you get poor quality oil. And the problem in the field is you don't really know what percentage of green seeds are there, because you have a brown seed coat, and these have to be used, put under a core sampler, and then they roll it down to find the percentage of green seeds. So normally 2% green seeds are tolerated, and frost can increase this up to 6 to 20%, reducing the value of canola by $15 per ton. So this is an estimate based from 2000. So the loss was around $150 million annually, and you can imagine after 20 years being in the prairie with unpredictable weather conditions, what sort of loss we are incurring even now. Right. So one of the things that we did was we started, in order to approach this problem, we decided to go back to Rhabdopsis, where we knew the fundamental genetics and found a mutant in which the embryo at maturity will stay green. And this mutant is called an ABI3-6 mutant, which is a null allele of ABA insensitive 3, which is an ABA-inducible transcription factor. And by doing a transcriptomic analysis at maturity, like this is around 13 days after flowering, and this is wild type Colombia, and we compared gene expression patterns in these two at 13 days after flowering, and looked at if anything stood out. And we were very excited that when we did this, the Mendel's green cotyledon gene, which is called as a stay green gene, or the SGR, really stood out, and it was severely downregulated in this particular background. So we thought, okay, maybe ABI3 regulates SGR expression, and that is required for degreening to happen. This work was done in collaboration with Dr. Fred Delmas and Dr. Peter McCord. And having figured this out, the next thing we want to do was, if ABI3 regulates SGR1-2 expression to control embryo degreening, and in an ABI3 knockout, you get green seeds fixed, what we should be able to do is we should be able to take this and overexpress SGR1-2 in this background and get the degreening to occur again. And that's exactly what we got. And you can clearly see this is Colombia, ABI3-6 with green seeds, and then these are 3 different lines with overexpressing either SGR1 or SGR2 under 35S. And clearly, when we overexpress that, all these lines had clear degreening happening, further demonstrating that ABI3 most likely regulates SGR1-2, causing degreening to happen. We've done a series of assays to confirm that ABI3 binds SGR1-2 to actually accomplish this. So very exciting story, and what we finally showed in this paper was the ABA regulates ABI3, and ABI3 can regulate the expression of SGR1-2, and that leads to embryo degreening, the process. And the ABI3 on its own can also regulate desiccation tolerance, seed maturation, and ABA sensitivity. So where do we go from here? So we were at this state And how do you actually move this? How do you make this into a useful technology? The first thing we did was we used Colombia and overexpressed ABI3, the master regulator of degreening, in this particular background. And what you actually see is when Colombia is actually exposed to frost, the seeds remain green at maturity, but ABI3 OX lines, even after being hit with -5 to -10 degree frost 2 hours per day, they clearly continue to degreen without a problem. So very exciting stuff. And based on that, what we wanted to do next was we wanted to move this technology into canola. And we recently published this work. Mendel Perkins is the lead author. It's interesting, Mendel, we are working Mendel's gene, SGR. Mendel was the first author in this story, and this was his master's work. And Mendel is at UBC now. So Logan and Neil also were instrumental in getting this story published, and where we actually showed that ABI3 from canola, when you overexpress this under a 35S promoter, you can get seed degreening even after frost. So just to go through the data, so here's a wild type canola line, West Star background, and 3 different transgenic canola lines overexpressing ABI3. They were hit with frost for 6 hours at -3 frost for 6 hours, and allowed them to mature. As you can clearly see, in addition to having a degreening of the seeds or embryo, the overexpression under a promoter like 35S also caused these lines to be frost tolerant. So you can clearly see West Star is hit pretty bad, and the overexpressors actually withstood the frost. And here's a picture after the frost exposure and following maturity. The seeds were sanded down and looked at the percentage of green seeds following frost. These are in triplicates. Wild type, following frost, you can clearly see the number of green seeds. And we had 3 different lines, 3 transgenic lines. And in this, L8 is the weakest expressor. And you can clearly see there's a little bit more of green seeds in L8. L6 and L7, overexpression of ABI3 led to elimination of all the green seeds even after exposure of frost. 2 different techniques were used. Either count the green seed percentage or look at the chlorophyll content in these seeds, and that clearly demonstrated that overexpression of ABI3 was sufficient to provide frost-tolerant degreening in these lines. So big question, is oil compromised? Oil is not compromised. I want to thank Raju Sula for helping out with these assays. Oil wasn't compromised. If anything, the percentage of oleic acid was significantly upregulated in these lines, in these transgenic lines, and the compromise was reduction in the omega-3 and omega-6. And similar to the oleic, we also saw an increase in nervonic acid, which is important for brain function, significantly upregulated, 20 to 30% upregulated in these transgenic lines. So another major phenotype that we also observed in these lines was the pod wall was thicker, the pods were stronger, and we also saw that rhiplum valve joint area was bigger. Right, it's larger in these transgenic lines. You can clearly see that it is larger in these transgenic lines. That leads to stronger pods and possibly prevents pod drop, or it could also lead to improved shatter tolerance. So we immediately looked at shatter tolerance from these lines, and I'll show you data in a second. So our pathway in the degreening pathway is this. So we have ABI3. And ABI3 regulates SGR2 to continue to degreen even after frost. And ABI3 can also upregulate these protective genes that can prevent and give the pods in the plant some frost tolerance. And also a misexpression of ABI3 through unknown regulators could be leading to increase in pod wall strength, and also the rhiplum valve joint area strength, increasing the pod strength, overall pod strength, and possibly lead to shatter tolerance. So we immediately tested shatter tolerance. This is Logan's PhD thesis work. And what we observe here is the wild type, compared to the wild type, which shatters very easily, these 3 different transgenics had very high shatter tolerance. And the reason we believe why this is happening is when normally under shatter tolerance, this is the endocarp. Anything in red is the Endocarp B layer, and that's highly lignified. And when the pod is drying out, the drying pod exerts tension on this Endocarp B layer, pulling the valves apart, exposing the seeds. And the ABI3 overexpresses. This Endocarp B layer is actually quite thickened. There's quite a bit of lignin, and it also bleeds. The lignin bleeds into the other layers, and we think the tension doesn't get transmitted to pop open the pod. So if you zoom in this section, this is the wild type. This is the Endocarp B layer, and in the wild type and in the ABI3 overexpressor, and you can clearly see these layers are drying out, and this tension, it never gets delivered to this layer that is responsible for opening up the pod. So Logan is finishing up his work, and he's characterized a number of genes that are involved downstream of ABI3 in the pathway, which we are not discussing here. So what we've shown is canola, ABI3 lines. There's no pleiotropic defects. There's frost-tolerant degreening. There's also improved shock tolerance. It's a very exciting story. We are definitely working with Nutrien to bring this technology to the market currently. And I want to take the last 2 minutes and also talk about a new program that's actually happening in the lab. So we call this the GAP technology, and we use this for gene editing for improved protein production in crops. And this work is from Logan's fundamental research work in the lab, and he's filed for a provisional now, and we're working towards getting a PCT filed and then in 2022. And what Logan was able to show was used to a specific overexpression of a specific gene. He was able to show that compared to Westar, the seeds accumulated at least 20 to 25% more protein. And what we've been able to do is, and this is a highly conserved process, we've been able to get the soybean homolog and rescue some of the mutants we have, and it functions very similarly. And so we do see a 20 to 25% increase in seed protein content. And what we are currently doing is, because this would be an overexpression technology, we are looking at negative regulators of this particular gene A. And once these negative regulators, we've identified a few negative regulators, and we are using CRISPR-Cas9 or the MAD7 system to knock down this negative regulator in order to enhance expression of gene A. That leads to expression of increased seed protein content. So we are in discussion with some of the pulse companies and to deploy this technology in their germplasm. We currently do not have a partner in the canola industry. We are definitely looking for some partners to work on this. So with that, I want to thank my amazing group of guys that are responsible for all the great work that comes from the lab, especially with the degreening work. I want to thank Mendel, Logan, and Neil for being for spearheading this project and getting into publication. So I really want to thank Raju for all the support he's extended with all the analysis. Peter McCord and Fred Delmas are co-authors on the paper. Thanks to Nutrien, ACDEF, and NSERG for the support. And so it's an exciting project. Look forward to talking more collaboration. Thank you for listening. I'd be happy to take any questions. Thank you.